Testing. Oh, great, you can hear me. I am Alec. I am the mentor from the Thomson Lab group. As Mitch mentioned, this is a group that was a spin out or kind of evolved from a SEAL group. So multiple high schools that participated in a Caltech program during the school year. Several of them from different high schools came together and now are working on the summer project that is still not done yet, and they will tell you all about it. Without further ado, let me take it from here.
Give a shout out to your teacher, Monica Barsiver, who's the coordinator of
Yes.
holding together this. And just one other point, what was one of the experiences you had in high school?
Oh, I was part of SRC, 10 years ago, so here we go. Great program.
Hello, everybody. Thank you for coming. We were gonna plug a thing.
Yeah, good luck making it work on that thing.
I don't think it has hello, everybody. We are the PFAS. Okay, finally. Hello, everybody. We are the PFAS annihilators, as you can see from our slide. We are a group of high schoolers aged 15 to 18. We are working under IEA, a Pasadena nonprofit, focusing on research and exploration. We are working with experts at the Thomson lab to develop a PFAS degrader and tackle the issue of PFAS contamination in our water. But first, I think we should introduce ourselves. My name is Camille Dahlgren.
I'm Lucas Garcia.
I'm JL Santos.
There's our faces. I'm Trey Reyes.
I'm Rick John. I'm Rick John.
So let's get this started. In recent years, the government and public have grown more and more concerned about PFAS chemicals, and it's with good reason too. PFAS, a kind of synthetic chemical such as PFOA and PFOS, have been found to interrupt the body's immune system and hormonal feedback loops, resulting in vaccine resistance, birth defects, and even cancer. PFAS can be found in just about anything, as stain resistant, grease resistant, and water resistant, ranging from your non-stick cookware to even firefighting foams.
The manufacture of these products leads to a PFAS isolated waste that seeps into the water supply. As you can see on the map, the light blue spots represent areas where PFAS contamination is above the EPA limits. The current EPA limit is 4 parts per trillion, and this is where our current PFAS detection technology sits. So this has spurred the race to create a PFAS as disposal technologies. Okay. The issue with this is this PFAS as a molecule is extremely strong, made out of almost indestructible all carbon fluorine bonds that take thousands of years to break. But here's a solution.
Focusing more into why we're focusing on a PFAS remediation method and we're going the biological route is that one of the current methods is filtration. Water treatment facilities use these 3 kinds of methods, but they only filter out the PFAS. They don't fully destroy it. The only existing, the one that industry mostly uses to destroy PFAS is incineration, which isn't really sustainable or good for the environment because it can create PFAS intermediates, which we cannot predict the effects it has on the environment or the body. Which is why we decided to go the enzyme route. Enzymes are far more sustainable, we have more control of the byproducts they create, and they are cheaper in the long run at scale. So our approach involves using a cutting-edge AI to produce novel PFAS enzymes. We plan to use a protein large language model. We plan to train this large language model off of naturally occurring enzymes that already degrade PFAS from literature. Then we will express these enzymes I'm sorry. So we plan to, so using the training data we get from, key fashion graders from literature, we plan to produce new and improved enzymes that will have a higher PFAS degradation rate to reduce that time. And we will then express those enzymes, and validate them on a PFAS substrate.
Okay, so now I'd like to sort of take a step back and look at our entire enzyme generation process as a whole. It has 4 interconnected steps. The first step is where we we fine-tune a protein large language model using existing enzyme sequences that are predicted to degrade PFAS. We then prompt the model to generate novel sequences and then do an initial vetting step. And we'll get more into that later in the presentation. So these generated sequences then flow into the second step, where they're tested for any PFAS activity that they might have. So any activity they might have when interacting with PFAS. And data that we gather from this module will influence future generation runs, essentially creating like a feedback loop that improves our system as we use it. We're also in this feedback loop starting to select for enzyme sequences that are more likely to have activity with PFAS. So any sequences predicted to have activity move on to the third step, where they're tested for experimentally and computationally tested to see if they're expressible in TXDL, a cell-free expression system. Again, data that we gather here will influence our future runs so that we're not only selecting for sequences that will have activity with PFAS, but will also be expressible. Here we'll also be expressing any proteins that are predicted to be expressible and sending those expressed proteins to Professor Ariel First at MIT, where she will be testing our enzymes on a PFAS substrate to determine if there's any catalytic activity or any observable degradation. And if we don't observe any degradation, then we have to alter our generation pipeline, potentially tweaking the predictions that we're making in the software that we're using. And if we do find that there is observable degradation, then we've effectively reached our goal of finding a novel functional PFAS degrading enzyme.
Great, so now that we have a sort of overview of how we're going to be generating these novel enzymes, let's take a look at just the first module. Anyway, so let's just take a look at this one right here. So a protein large language model essentially just produces coherent protein sequences. And the way it does this is by masking amino acid sequence or amino acid within a protein sequence. You can see here that F is masked and A is masked. Masked basically just means hidden, and the large language model is then made to essentially guess what the amino acid is based on the other amino acids within the same sequence. So F here and A here are the most probable out of a set of F, W, and L, and A, V, and L, and they are picked based on those probabilities. And from here, we can essentially make coherent protein sequences based on just this training data. So in our process, we're going to be using base protein LLMs that can already make these coherent protein sequences. So we're using 3. ESM2, ProGPT2, and Zyme Control. So these protein large language models are already trained based on protein sequences that they find on the internet. And from here, what we're going to be doing is that we're going to be finding enzyme candidate sequences in order to fine tune these protein language models in order to produce biased protein sequences that will be able to degrade PFAS. And from here, these enzymes that we produce will be going through a post-generation vetting system where we will be essentially filtering out any erroneous sequences that don't really make sense, or filtering out any protein sequences that aren't able to really fold, or any protein sequences that are just too unnatural to be folded at all. And from here, this goes on to our second module, which is this one.
Okay, so taking a look at our second module from the first one, like sort of look under the hood here, and there's actually two internal mechanisms that make it function properly. And so the first of those mechanisms is a PFAS binding classifier. So this classifier essentially takes in our generated proteins and predicts whether they will be able to bind to PFAS. Again, that's sort of where that feedback loop happens with the first generation process. And any sequences predicted to bind to PFAS will then move on to the second mechanism within this module, which is a PFAS docking step, where we take the simulated three-dimensional structure of the generated enzyme and simulate it with a PFAS ligand, like for example, PFOA. And we'll sort of gauge the affinity there computationally. The data that we get from this step will influence the PFAS binding classifier, essentially allowing it to make more accurate predictions and giving it more training data to go off of. So this is sort of another secondary feedback loop within the module that improves the module as we use it, making our predictions more accurate, hopefully. So moving on from this step and then going into the third step, there are also actually two internal mechanisms at play here. And again, similarly to the last one, another classifier, but in this case, it's an expression classifier. So this expression classifier takes in our generated sequences and predicts whether they will be expressible in TXDL, or the cell-free expression system. And then again, that feedback loop with the first step, selecting for sequences that are more likely to be expressible. And any sequences that are predicted to be expressible move on to the expression validation step, where we'll be actually taking these sequences, ordering the DNA and then expressing them into XTL. And whether if they express successfully in lab or if they don't, that data will be gathered to allow the expression classifier to have more informed predictions, more accurate predictions. Again, creating an internal feedback loop, improving the module as we use it.
Of course, the AI requires ERS training data. So in order to get this, as we did an extensive literature review and found on 3 enzyme families as we predicted to work for PFAS segregation: peroxidases, ligases, and dehalogenases. Now, peroxidases and ligases as produce radicals that stabilize the structure of PFAS, and but they are not very specific. They can affect other molecules and potentially become toxic in of themselves. Meanwhile, the halogenases have specific active sites that target PFAS and PFAS-like molecules, allowing us to target as these molecules more active.
After further literature review, our team identified a promising halogenase candidate, enzyme A6-RDHA. Enzyme A6-RDHA is found inside Acetylmicrobial species shrimp A6, a type of soil-dwelling bacteria. When this bacteria is placed on top of the PFOA substrate, it's been found over time the concentration of PFOA decreases, whereas the concentration of shorter chain PFAS increases, showing direct evidence of enzyme A6-RDHA's involvement in PFAS degradation. That kind of exact evidence made it distinct amongst other dehalogenase candidates, and is why we chose to focus on it. However, there was an issue. Enzyme A6-RDHA has only been experimented on inside the microbe. Scientists have yet to fully characterize it by itself and provide a complete protein sequence, so we're only able to obtain the fragments. Thankfully, this fragment was enough. We ran the fragment through FoldSeq, which is a program that compares your inputted protein sequence to other protein databases and provides structurally similar enzymes.
So the reason why this is important is because with structural similarity, we can also assume functional similarity, which is an important aspect of finding valid training data. So we found these 68 natural enzymes. These are just 2 examples, and we just know that these are good. But there's another problem. We also have to make sure that these enzymes are sequentially diverse. So on top of them being structurally similar, we also need them to be sequentially diverse And the reason being is because if we have protein sequences that are too similar to the original enzyme that we already have, then we will not produce any novel enzymes, meaning that we'll just get the same protein sequence over and over again. And that's not what we're looking for. So in order to showcase our sequentially diverse enzymes, we created a heat map that looks like this. So the way that we created this heat map is just by aligning the sequences together and just by comparing the two. So you can see from here, that's the technical aspect, and this produces this. The way to read this heat map, if we just take a look at just one square, this is comparing two enzyme or protein sequences to each other. And what this produces is a similarity score. This one in particular produces 37.28%, which showcases like a darker hue. And if we put this into context of the rest of the heat map, we can see that this color is shown throughout the heat map, showing that our enzymes are sequentially diverse, which is good. You might be wondering, like, what's this bright line right down the middle? That's to be expected because that big like bright light right down the middle is actually the enzymes being compared to themselves. And when you compare anything to themselves, you can expect a 100% similarity score. So, great.
Okay, so earlier in the presentation, I talked about an expression classifier. And you might be wondering, like, why do we need an expression classifier for these generated proteins? And the simple answer is that generated proteins are incredibly difficult to express in any medium. Here's just one example with generated PETase variants that are, so PETase is a plastic degrading enzyme. And we generated 6 of them, well, 6 standard, found 6, and then we started working with those, and we wanted to express those in a self-free mixture. But only 1 out of 1 out of 6 were successfully expressed. And that's actually pretty good as for like generated proteins as they go. But when you keep in mind that these were incredibly similar to like pre-existing PETase variants, it starts to get in the context of the issue itself, which is the farther you get from natural sequences, the more difficult it becomes to express them successfully. And we want novel proteins. We want to explore new structures. We don't want them to be exactly the same as natural ones because we're not really, treading new ground. So that's where that classifier comes into play, where we are inputting a protein sequence, a generated protein sequence, and are getting out a predicted label, like for example, expressible, and then the probability of that label being correct. And that's really helpful because you don't have to go through the pain of like going in lab and expressing a bunch of proteins and saying like, oh, that one didn't express properly, or yes, this one did. And this sort of lets us streamline our process. So there really isn't an expression classifier out there in the world that is accurate enough currently. And so we decided to actually build our own classifier, gather all the data necessary for it. And so our plan is as follows. We're going to be, we're generating 2,000 sequences per large language model, protein large language model, essentially allowing us to sample like a bit of the possible structures that these models can produce. And then taking these generated sequences and putting them into DNA sequence libraries, ordering these libraries, and then taking this DNA and actually expressing all of these generated proteins in the cell-free mixture. After this, we take the expression solution and use something called a technique called protein mass spectrometry to determine which proteins were successfully expressed and which were not expressed in the solution. This way we can sort of gather data to see what structures or what sequences are getting expressed and which aren't, and hopefully finding a pattern in that data. And then we input this data into an architecture, a classifier architecture that we will sort of find the most optimal one. That classifier will hopefully begin seeing these patterns in the data and begin being able to predict what sequences will express and which won't. So currently, we've actually started, we don't actually have the training data yet. We're actually currently doing the mass spectrometry step on it as we, well, not as we speak, but we've already submitted the samples. We've just had some practice with the classifier as we're waiting for those samples to get, run and analyzed. We've sort of collaborated with the Human Interactome Project here at Caltech, and they've given us 564 human receptor protein sequences, and they've expressed all of these, given them a qualitative expression label, being either undetectable, faint, low, medium, or high. So using this data, we tested out a couple of different classifier architectures, and we landed on this one, which was a multi-layer perceptron, and we also combined different labels together into actually 3 bins. So we combined undetectable, faint, and low as one bin, and then left medium and high on their own. We were tested out a couple of different combinations of bins. And the overall accuracy was 75%, which wasn't the greatest, like the highest accuracy, but we were also basing it off of how many false positives and false negatives it was producing. And this one had the lowest false negatives, false positives. It doesn't do great at detecting or predicting mediums, but we are sort of more worried about the highs. So that's sort of that's what we want. We want high expression. So there is sort of a caveat to this, and that is that the data we use to train it is, because we blend it together, is incredibly imbalanced. So that can become sort of an issue where you worry about bias in your model. You worry about that it's only predicting, it's only choosing like the most. So there's a bunch of undetectable faint and low data. So all it has to do to get a high accuracy is just predict that every single time, which is a worry because we want them to find patterns in the data, not just sort of take the easy route. So yeah, that's where we are so far.
So I know that was a lot, but I just want you to remember 5 things to take away from this presentation. First, our drinking water is contaminated with a dangerous chemical called PFAS. We are working on a 4-step interconnected enzyme generation process to develop a degrader. We have 3 enzyme classes for our training data, and out of that, A6-RDHA is our most promising degrader, and we are building our own expression classifier. Next, I just quickly wanted to talk about iGEM. As I mentioned, or as Keith mentioned at the beginning, we are an iGEM team. We are looking to go to Paris in the fall, but first I just want to talk about a little bit what iGEM is. iGEM stands for Internationally Genetically Engineered Vaccines. It is the premier international synthetic biology competition for university, overgrad, and high school. We will be competing as a high school team in that division. And iGEM pushes innovation in science and engineering. So we want to go to iGEM, basically. I think Jail's gonna talk more about fundraising.
Well, submitting our work to iGEM is actually a very expensive endeavor, and we would really appreciate any advice anyone in the audience may have on how to raise the money for this. We are currently advancing towards a $25,000 goal. So even a direct contribution would be great, because you'd be assisting in improving your environment and our communities. So here are our acknowledgements. I want to say thank you so much to Ms. Monica Barsever, Alec Lorenzo, Zach Martinez, Dr. Galia Barsever, Melody Wu, Anna Mikova, Nicole Endicott, Professor Matt Thomson, Dr. Woj, and Professor Kaizen, and Professor Ariel First at MIT for their help and support. Thank you. Any questions?
Questions? We come back from astronomy to Earth and real world problems here we're dealing with.
You mentioned that you use your code to sort through the generated proteins from the models, and then you use libraries to like categorize that. What language is that in? What libraries do you use?
Okay, we are coding in Python, mainly. Well, for the libraries, I mean, we're generating, do you mean like the code packages or something?
Yeah,
that we're,
you mentioned that you used 2 libraries, but you didn't say what they were.
Oh, sorry, I might not have been clear. The libraries are of generated proteins that we're creating. That's sorry, yeah, it's kind of confusing. But we generate proteins and put them into libraries, and then get their DNA, but anyways, yeah. For the packages for the code, I'm not sure, I mean, there's a couple of different ones. I don't know, you guys remember? I'm not totally remembering them like off the top of my head, but yeah, I mean, I can get back to you on that one.
Is the code base available for this?
Yeah, we're working to make like a easy to use on Google Colab, yeah, so people can use it and check out our classifier and stuff and our generated proteins, but yeah, so, yes.
So you guys are introducing an enzyme to break it and drinking water, are you sure that the enzyme is upset to do this?
Well, I mean, the.
So, we're not really at that stage yet, but of course is something that we are considering. We're actually we're considering putting it in, wastewater treatment facilities as they have, actual enzyme pools that they already use. And from there, then we'll help affect the water supply. But as we develop it more, we test more, we'll figure out whether or not it's safe, but hopefully it will be.
What are like the general, I mean, this is more long-term, but once this enzyme has been fully developed, what are the stages that it would have to go through before wastewater treatment plants have to actually like start using it? What's the protocol?
So I will be honest, I'm not entirely sure yet. We'll totally, absolutely be looking more into it as that is, a good requirement for iGEM. But most likely it might be approved, have to be reviewed by, more official bodies. That's all I'm gonna say. I don't want to inform the public. I want to say the EPA, but I'm not entirely sure. But it's something we are considering. We're just not there yet.
Yes.
So the PFAs, 1,6 goes to like these enzymes, they degrade into smaller molecules, of course, but is it more that it degrades into smaller polymer chains of PFAs, or they degrade into totally like benign molecules? It's more like, are these smaller chains more likely to degrade naturally that don't take 100 years?
Yeah, it's these smaller chains, there is sort of a worry, like you're just breaking it down into smaller, like more problematic molecules, which is definitely something you need to keep in mind, but yes, when you break them down, they're more accessible to microorganisms to break apart as well. We're trying to make sure that our enzyme breaks it to like up to a level that it can be easily broken down over time, but there is a worry that like, if you just pop off like one fluorine on it, it's not really helping anyone. But yeah, that is something we're thinking about, and definitely a consideration for like for the enzyme design as well. Yeah.
I could also expand on it further. One of the reasons why it's so difficult to degrade naturally in the environment is that the length of the carbon fluoride bonds within these molecules don't occur naturally in nature. If you get it around to like 3 carbon fluorides, I don't remember the technical name, but at that point there exist microorganisms that can break that down. But yeah.
But would those molecules still be harmful? Like if you were to, again, going back to like some sort of treatment plant where they break these down, if that then becomes drinking water, are there still those micro plastics in the drinking water that's just smaller?
So I will say that, okay. Hold up, give me 1 second. So most of the information that we have, well, most of the ones that have been studied are PFOA and PFOS. Because, I don't know if we made it clear in the presentation, but this is a category and there's 15,000 different kinds of PFAS. But most of the studies of the medical impacts have been done on those 2 legacy molecules. So if they do get further broken down, we would call those, PFAS intermediates, and we aren't entirely sure of the impacts on the body just because we haven't directly studied those. But the assumption is that yes, it would still be dangerous.
Okay.
Is there any thoughts on when you get the right enzyme, how you would max produce it? Would it be cheap enough to use in everyday life?
Yeah, I mean, from what I understand, like at a smaller scale, it's pretty expensive to create proteins in general. But if you have like a sort of streamlined process at scale, it gets pretty cheap, especially comparing it to incineration, which is the alternative. It would probably be a little bit more economical. But yeah, I guess that answers your question. Yeah. Yes.
I just wanted to ask, if you break down the PFAS and then they become the intermediates, and if that's still dangerous, what's the end goal, like to just get rid of it completely?
Yeah, our goal would be to break it down more totally, not to those only smaller intermediates. We want to break it down as much as possible. And I've seen, I don't know, I mean, if you guys want to correct me, but I've seen stuff where smaller change are definitely less harmful to our bodies in general. I think so. I don't, maybe I shouldn't be saying that, but I'm pretty sure we talked with experts, I think they discussed it, but there are unknown ramifications of doing that. Yeah.
Is the end goal to break it down to the atomic level?
It would be nice to completely defluorinate it. Yeah, I mean, that would be probably the ideal, the goal, probably, yeah. I don't know if that's possible necessarily. Yeah. Yes.
Why mechanistically, like, why are PFAS harmful? What is the chemical?
Yeah, okay. So, well, you know how like a cell is made with like the phospholipid bilayer, and well, mechanistically, PFAS is made up of kind of these like they have a weird structure that makes them so they bind to that like that phospholipid bilayer, and then it makes the cell essentially like it makes the cells like weird. What I mean by that is like it lets stuff in that's not supposed to go in, and it also potentially like essentially lets like the cell guts out, which makes it so that like once the PFAS is in like your cell, it's not great for you. It lets all the stuff out, that's bad stuff in. But that's the simplistic overview of that. Yes.
Is the PFAS itself directly harmful, or does it just let other stuff in? And like, does it do damage to anything other than the phospholipid bilayer?
I'm not sure. Not necessarily. So there was actually this one there's this one study, I think it was in like North Carolina, I think. A mother was exposed to it, and then the product of that was that the baby was massively like a lot of deformities, and the reason being is that a lot of like the very simple answer, no, like PFAS by itself is not intrinsically harmful, but the stuff, but what it does to your cell, what it allows in, and the stuff that it gets out from it, that's what's more harmful.
And can it spread, like if it goes into one cell, can it spread to other cells?
Yes. Yes.
Would there be like a potential treatment if you're exposed to like repair the phospholipid bilayer? Like that could ever be.
Like an anti-PFAS?
Yeah. It's not actually doing damage out of it, just to layer.
Yeah. Okay.
I want to say that right now you're really seeing the division of our team. So I human practices, so I know most of these more general questions. I want to step back to the question about the toxicity of PFAS by itself. By itself, it is pretty toxic. It has the fluorides, which if it does end up, splitting off, it's an acid that's really bad for you. It also can have the potential to rip open the cells. It's mostly the fact that it's ripping open the cells, it's clogging the cells, it's interrupting the endocrine system. That's how it's able to form all those cancers. I will also say for the next question, that's a little bit out of our ballpark because it's getting more medical, but we are in contact with a college iGEM team that's addressing that issue. Repairing the biolipid bilayer, phospholipid bilayer, I wouldn't really say we can answer that. That'd be pretty cool if it was out there. We're mostly a degradation method.
Thank you. Great question.